Lovely, thanks. Yeah, thank you.
We're a little ahead of schedule.
Sorry?
We're ahead of time.
So maybe I'll just say the nice things first before I get to the talk. Yeah, the nice things I wanted to say. Big thanks to the local organizers. I think this is really a very pleasant ISMB. For me, it was the first ISMB after Corona. And for example, I can remember ISMBs where you had to queue ages to get your lunch. This is all organized so efficiently, and the conference center is very nice. You step outside, you're in the middle of town. It's really a joy to be here. And the second thing, maybe, while I still have some minutes to kill before I'm allowed to start with the contents, this is work that I've done together with a PhD student of mine, Ali Al-Fadlawi, who unfortunately cannot be here because he had visa problems. So we had to organize all of this pretty last minute. And they're also thanks to the organizers who got this sorted out on the last days towards ISMB. It's okay to start? Yeah. All right. Good. Okay, so let's get going. The title is maybe a little bit innocent. Can proteins be represented through secondary structure? But you will see in a minute that I think it connects quite nicely to Jana's talk this morning. Yeah, she's looking surprised. Because this is all about a really distant evolution. We will see on the very last slide, archaea, bacteria, eukaryotes, proteins that are structurally pretty similar, or you can see the similarity, although in terms of evolution, really very, very distantly related only. And it also connects to Martin's talk from yesterday, which, by the way, I'm really, really happy for you and your work. I'm a big fan of FoldSeq and then all of the other work that you're doing. And it connects in two aspects. I was surprised to learn about the important role of the big data in the metagenomic, which I think clearly shows the huge variety of sequence data that is there and that is useful for prediction. But it also connects to something more profound on FoldSeq, and that is how to represent sequences. And I will give you here a very tricky thing. One example, but it's one where I think where sequence completely fails, but also what we do right now with structure has its problems. So let's get going. And these are single-strand annealing proteins. So these are proteins that an experimental colleague of mine in the institute that I am is working on already for 2 or 3 decades. And single-strand annealing proteins are very fundamental to all kingdoms of life. They're important in DNA repair, and they can basically take single-stranded DNA and find the fitting base pairing counter strand to match. And so that is obviously very important. In human, there is a single-strand annealing protein called RET52. It plays a role in cancer. It's an important cancer target. But the scientific community has also studied these proteins in lambda-phages, viruses that infect E. coli. And also in E. coli, you can see single-strand annealing. And here on the right, you see how this is experimentally determined. So under the microscope, you can see the resolution is a bit poor. But here in this abstraction at the bottom, you can see this is the double-stranded part. This is the single-stranded part. And these big dots that were fluorescently labeled, that's the RAT52. And as I said, there is already, even without any structural insights, which were difficult to get by, there's already interesting stuff about these proteins. About 15,000 or so of them are known right now. 90% are bacterial, 9% are eukaryotic, and only 1% are archaea, but they appear in all kingdoms. And 1% may seem low, but if you look at the whole of UniProt, then about 2% of sequences in UniProt are archaea. And I think that points to two things that I think both Jana and Martin said, that in terms of the representative of the sequences and the data that we have, it's pretty skewed. And I think that's very important for maybe those bioinformaticians among you who not so much in the wet lab, The reason why it's skewed is that these archaea are much more complicated beasts. They are difficult to cultivate, and that is the reason why we know less about them. It's not because they're less important or more boring or something. So single-strand annealing proteins, this beauty over here was a structure that was resolved about 20 years ago, and for 20 years this was the only structure in that class of proteins. And I find it really super beautiful. If you see it in 3D and can fly around, it really looks like a spaceship, a donut. In red, you see the positions where the DNA is binding. So the single-stranded DNA winds around it kind of in a circle. And yeah, and this structure poses, I think, really some big challenges to search, even if you do have structure in other proteins available. And that is, the inner lining of this ring is very important to form the ring structure. But you have a part that is pointing to the outside, because this ring, I should say that this ring is composed of 11 units. And the parts that are pointing to the outside are evolutionarily not constrained at all, right? You can really make extensions to this just to create a star structure, if you will. And for those parts that point to the outside, no constraint whatsoever. And that means there's no sequence matching, there's no structure matching, and it actually doesn't really matter. But the inner lining is the part that is important. So basically for 20 years, there was only this structure available. And in terms of sequence, these 15,000 single-strand annealing proteins that were known, they broadly fell into 5 classes. And in the community, there was a huge debate. Is that like one family? Is this all completely independent? How does it relate? And as I said, there was no structure. But of course, we all know today there is structure, or at least predictions. And today we have an answer. And this is the key slide for Martin. Because I think when you look at these structures, you see the similarities, right? By eye, you can get them. And the similarities is essentially that there are 2 motifs, this beta sheet motif with a perpendicular helix, and then there is this beta hairpin, which is the second motif. The beta hairpin is less conserved than the sheet motif. And the variation you see here, and the 2 proteins that were so much under debate are the human RAD52, which is here, and this is the E. coli red beta. And you can see the strands in the sheet are much shorter, the helix is much shorter, but it's still there. There's kind of an additional loop, and the loop structure is there. And basically, along the columns, no sequence similarity whatsoever, nothing. Within the columns, some sequence similarity, but even there you see in terms of structure quite a bit of variation, so that when you superimpose them, it's not that easy. And you must know also that this variation is about 100 residues and 250 residues. So this is flanked by stuff that is not conserved at all. So I think a key problem in all of the searches is how to find the part that is really nicely conserved, and then how to set the threshold that you still accept that this is variation which is conforming versus not that seems to be random. So back 10 years, this colleague of mine, he published this, and I found this really funny, I must say, because I thought that was pretty misleading and pretty stretching it. So on the top, you see a multiple sequence alignment of RAD52. At the bottom, you see multiple sequence alignment of RAD beta. What I find misleading is the yellow color, which he put, which kind of, you look at it and it looks as if there was conservation, if yellow means conservation. But if you look a little bit closer at this figure from his paper and you look at the actual characters, then you see, well, there is actually within the family RAD52 and RAD beta, there is of course conservation, but between them, nothing. And in fact, in black at the bottom, you see that is really just a handful, 2 glycines and a tryptophan in 100 residues. It's really just 5 residues or so that you could argue are conserved. So I thought that is mad, to say they are related. However, there was something that struck me, and this is really one of the most highly esteemed colleagues I have, and that is that, right? That although the sequence similarity is not there, what they do have in common is the secondary structure similarity. They both have this pattern. And I thought 10 years ago, even without all of that AlphaFold structure, what he was missing, and unfortunately at the time I was not cooperating with him on this, what he was missing was just the ability to search by secondary structure and the ability to give a statistical significance of a finding. How likely is it, or how random is it, that you find strand-strand helix, helix, strand-strand helix? Right? Is that likely? Do you find that often or not? And the answer would have been, you don't find it that often. Of course, a single helix, as Martin also pointed out in his talk yesterday, a single helix, easy, but that conjunction, difficult. So what we've been doing is basically we rendered some results on representing structure by secondary structure rather than tertiary. And what we found is that for superfamily classification, we could tweak it that secondary structure was nearly as good in terms of classification of superfamily membership as tertiary structure. And of course, it was much, much better than sequence. And the beauty is that secondary structure is suitable for sequence algorithms. So you can just take your standard BLAST. And in contrast to tertiary structure comparisons, it can handle the structural flexibility, this bent a set of sheets, this bent set of strands that matches a straight set of strands. And so there's a slight edge. And I think this problem of how to make use of these topological features in search is really an important one, because we try to quantify how important in remote homology structure and maybe these advanced comparisons are in terms of sequence. And we systematically did an all-by-all comparison of sequence similarity to structure similarity. You know these plots, but we quantified also the size. And basically everything that is here on top is the stuff that you try to find, which you could argue are remote homologs. And by sequence, you can find about 26K. But by structural similarity, you have in this kind of subset, this benchmark subset that we did, 1.1 million, right? This is what we're missing with sequence method and what methods like fold-seq, etc. can give us. But this must be offset against the stuff we don't want to find, 61 million, right? And there we have to be careful that we don't dip into this. And so to conclude, our preliminary results suggest that these really hairy cases, I think that must be also highlighted. This is really just when you go from bacteria to human, that their basically secondary structure can be very, very helpful to complement tertiary structure, and in particular handle this hairy problem of flexibility. So the answer is yes. And in this animation, you see a selection, I think, of 3,000 out of the 15,000 proteins in terms of their structural similarity on this one motif of the strands aligned. And if the color changes later on, then they belong to different families. And I think you will agree that you always clearly see these things should be together from a structural point of view. But you also get a visual impression for how difficult it is, even with a kind of a vectorized structural similarity feature, to capture this degree of flexibility that the individual changes show. And yeah, if you're interested in further details, you find at the bottom 2 papers that we published on this. And if you're interested to work with us, then please send me an email.